Okay, so our experience is parceled into events. What I'm going to show you now is me giving a talk about this project a while back to my lab members. It was one of the previous formation of this work. And this is Alon. He's part of my lab, and he's trying to make sense of what I'm saying. Now, let's assume that this sound wave is me talking. So you can see that it's continuous, but sometimes I speak and sometimes I stop speaking, probably between slides. That's when I stop speaking. And although this sound wave and this experience is continuous, according to the event segmentation theory, your brain parcels this experience into chunks of events. For example, at the end of slides. So let's say that I start with slide 1 with the title. At the transition between the first slide and the second slide, when I stop speaking, there will be an event boundary. And then a new event will start. Let's say that this is slide 2, intuition to the big problem. And again, when I switch slides, there will be an event boundary. And maybe someone will ask a question. That's another change in context that will tell your brain that a new event needs to be formed. At the end of the answer, there will be an event boundary. And then I'll introduce some literature review, an event boundary. And on slide 4, I'll tell you what's still missing and how I propose to solve the problem. Now, what your brain is doing during this continuous event is to parcel this continuous experience. And it is believed that once an event boundary is found, when the context of the experience is changed, your brain just gets the gist of the event that has just ended and stores it into memory. And this is how we create new memories as we go through our daily lives. But isolated event representations won't help you make sense of your experience, right? So in slide 4, when I'm telling you what's still missing, you need to know what is known. And what is known was presented in slide 3. And when I say that I'm going to propose a way to solve the problem, you need to know what problem that is. And the problem was introduced in slide 2. So we need to go through our lives and making inferences about what's currently happening and how that relates to what we know and to previous events. So I wanted to know how these relations between events are being formed. So I'm going to present a few clues that helped us to create this project. And the clue number 1 is that there is replay at event boundaries. So replay is the sequential reactivation of cells, and it has been found in rodents, typically during rest or sleep, after navigating a maze. So in the hippocampus, we have play cells, and these cells basically code the space. So when you're in a specific location, say this red spot, then one cell would fire, probably more, but in this example, one cell would fire. And then when you move to the pink area, then another cell would fire. And when you move through a path, then you have a series of cells that are firing, indicating where you are in space. But it turns out that when rodents are resting or sleeping and they're not traversing any path at all, you can see these rapid reactivations of the same cells in the same order, representing the transverse path. So it is commonly believed that this replay is building their knowledge of space. It's rehearsing where they have been and where they're going to help them understand where they are and understand the space they're in. Now, we also know that there's reactivation in the human brain. And the reason we call it reactivation and not replay is that we don't see this sequential reactivation of cells because we don't have that the resolution to look at individual cells when we're using neuroimaging. And this specific study, it's an EEG study. They used the Sherlock movie, the BBC Sherlock movie, and they found that at the transitions between events, and events here are just scenes in a movie. So when there is a cut in the movie and a new scene begins, then they see the reactivation of the just ended scene. So to give you an illustration, if this is again my sound wave, it would mean that at event boundaries, there will be a cut. At this cut, there will be a replay of anything that happened during the just ended scene or event, okay? The only difference between the animal work and the human work that I presented is that the animal work is mostly seen offline during rest or sleep, but the human brain actually does this replay or reactivation as we experience things. So it is online. In both situations, both in human work and rodent work, the common hypothesis is that the function of this replay is to chunk events up and store them into memory. So again, there is an offline function to this replay. So let's understand what this means. I have just switched slide. At this point, your brain would have an event boundary. It would sum up everything that has just happened in the previous slide. So it would tell you, for example, that there's replay in rodents during contextual shifts in an ongoing experience. I also said in the previous slide that there's reinstatement in humans during shifts in an ongoing experience, and that both phenomena are presumed to have an offline role. So this is what should have happened in your brain right now. Now, interestingly, event processing and replay also engage the same brain regions, specifically the hippocampus and the default mode network. The default mode network has been mostly believed to be an offline network. When you do some engaging task or a sensory task, some areas of your brain, mostly sensory, will be active, and then you would see these blue areas. And in fMRI, all activation is compared to a baseline, and usually that baseline is rest. So areas that are active above a resting baseline during task are those that are active in helping you do the task. But these areas that belong to the default mode area actually reduce their activity during the task. They increase their activity during rest. And this is why people believe that this network is in charge of offline processing, like envisioning the future or remembering the past or just mind wandering. However, these very same brain areas of the default mode network have been implicated in event processing. So for example, when you're listening to a story or watching a movie, these areas actually increase their activity at event boundaries. Furthermore, another study found that they actually hold these gists of events. So if people watch a movie or just hear the movie or tell about the movie to a friend, then the same brain areas would hold the similar representations, both for the movie watching and for telling about the movie. Now, if you think about it, these are very different experiences. When you're telling a friend about a movie, then you don't see the movie, you don't hear the sounds. It's your internal impression of the movie. It's the gist that you're telling about. And this gist is also apparent when you're watching the movie in these brain areas. And this is why it is proposed, these areas are proposed to hold an abstract event representation. I should also say that there's a line of studies that use inter-subject correlations to study these phenomena. So what they do, they actually correlate the brain activity between brains of different participants. And I'm going to touch upon that as we move on in this presentation. Now, these studies that I told you about are related to the processing of events in humans, but we know from rodent work that these areas are also related to replay. So in this study, this is a MEG study in humans, you can see this line, this vertical line representing replay in the human brain. And they asked which brain networks across many brain networks are mostly activated when this replay occurs. And they found these two networks, the red one and the blue one, which roughly correspond to the default mode network. In animals, specifically in mice, they found that when slow wave ripples occur, and this is when replay also occurs, then the area that is most activated is the retrosplenial cortex, that in the human brain that in humans would correspond to this precuneus retrosplenial cortex here, again a region of the default mode network. So this is just to tell you that another clue that we have here is that event processing and replay actually activate the same brain areas. There is a link between these two phenomena. Clue number 2, there is offline replay that links related events. So I want to tell you about a rodent study, a very elegant one. And here it was rats, I think, and they transversed through a maze. And they could either go in one compartment of the maze. I hope you're seeing my mouse, so it's on the left side, or they can go on the right compartment of the maze. What they never ever do is go through the entire outline of the maze. It's part of space, but it's not part of space that they have experienced. And when they sleep, then we can see this replay again. We can see cells firing in a certain order that actually maps to the one compartment on the left and the second compartment on the right. However, sometimes you can actually see cell firing that represent the entire contour of the maze. So although the rodents have not experienced that route in the maze, their brain built an internal model of the space. They understand that these two compartments can actually join together to form the big maze. So it is as if it is piecing together different parts of experience to form an understanding of what has gone on, of the world around us. And if this is true, then maybe this online replay that we see in human can link related events to help us understand our experience, to create an internal model of what we experience in the world. So in slide 4, when we need to form links to slide 3 and to slide 2, it could be that these reactivation of previous scenes will help form these links. So what we asked in this project is temporally remote information, right? Not just the just ended scene, but actually information that we gathered before that. So is temporally remote information also replayed at event boundaries of naturalistic experiences? And by naturalistic experiences, I mean mostly narratives, and I want to explain why. Oh, but before that, I need you to understand the hypothesis, right? So again, I switched my slide. You should have an event boundary, but my hypothesis actually says that here, in this event boundary, your brain would replay not only the previous slide that I presented, it should replay any relevant information that I told you so far in my presentation that is related to this hypothesis. So this should happen very rapidly in your brain. It would say this, well, the brain parcels ongoing experience into events. That was slide 2. There is online replay of the current event at event boundaries, slide 4. But to make sense of ongoing events, temporally remote information must also be replayed, slide 3. Offline replay can do that, right, in rodents. So can online replay link remote events in humans? So our experimental approach, narratives. So far, people studying replay have used controlled lab design experiments. And besides the disadvantage of not having the ecological validity of representing our real life, another problem is that when you learn a maze or when you do a controlled task, then you repeat the task again and again and again. So the representations of events would be identical across repetitions. Now, how can we study how unique events are bound into an internal model if we can't spot the relevant events? They are identical copies of each other. The advantage of narratives in human studies is that each event is unique. When you watch a movie or when you're listening to me now, everything that I say will not repeat again in the exact same way, right? So we have unique events and they should have unique representations in our brains. So to use this paradigm, we use 2 freely available data sets from the internet. The one, and they're very, very different from each other. The one is called the 21st year data set. We had 25 participants listening to an audiobook. So it's only auditory. The second is the Sherlock movie that I told you about. People in the fMRI are watching the movie and they've never watched this movie before. So this is audio-visual and this is only auditory, but there are many more differences between these 2 stimuli. For example, the content, one is drama, the other is mystery. The narrative structure. So in the Sherlock movie, each event leads to another event. So it's a linear arrangement of events. But in the 21st year, they had 2 unrelated narratives, story A and story B, and they're interleaved. So participants are listening to scene 1 from story 1, and then scene 1 from story 2, and then Again, story 1 and story 2 and so on. This goes on for 30 scenes, and on the last 15 scenes, these two stories merge. But before that, they involve different characters in different locations. They're completely independent. The stimulus structure is also very different. So in this story data, the scenes are of very same length. They are about 1 minute. So you can see the distribution of lengths is quite narrow. For the Sherlock data set, it's all over the place. It's naturalistic, right? It's a movie. So some scenes are very short, actually quite a lot of them. Some are very long. And this introduces some difficulties in our analysis that I may touch upon later on. The pre-processing is different. So I downloaded the story data set from the web, fully pre-processed, but I used my own pre-processing for the Sherlock data. And the reason I'm telling you all this is that there are so many factors differing between these two stimuli that if there is any consistent effect across the data set, this will tell us something general about the neural processing of naturalistic stimuli. We can ascribe our findings to either of these very different factors across the two data sets. Okay, so experimental methodology, representational similarity analysis. So this is what we do. We have our movie. Okay, let's say this is the movie. And then in the transition between scenes, we have an event boundary. And we know exactly when this happens because we know the exact second of the movie where this occurs. And then we find the TR, the smallest unit of acquisition that we have in fMRI that contains this event boundary. And in these experiments, each TR, each unit is 1.5 seconds. Now, the problem is that the bold signal that comprises all fMRI measurements is quite sluggish. So when you are experiencing this event boundary at a certain time, the brain reaction to this event boundary will come only 4 to 6 seconds later. So what we do, we take this TR that contains our event boundary and we shift it with 4 TRs, that's 6 seconds, and that should be around the peak of the bold response. And now we take all our event boundaries, EBs are event boundaries, event boundary 1 between scene 1 and 2, and event boundary 2 between scene 2 and 3, and so on. And we take a sphere across, so we're going to look across the entire brain. And our brain is divided into voxels. The voxels are like 3-dimensional pixels. And we take a pixel and we take a sphere around it. And for that sphere, we're going to take the representation. And what is a representation? If we have voxels here, each of them has their own unique activity level, which is represented here in different colors. This is just toy data, okay? So this is not real. This is just an illustration. So this representation is the voxel activity levels of all the voxels in the region I'm looking at. So obviously in each event boundary, I will have a different representation. What I also want is the representation of not only of event boundaries, but also of events. And events are scenes. So I'm separating my event boundaries from my scenes by 7.5 seconds, again, because this bold signal is sluggish and I want to separate the signal of the event boundary from the signal of the scenes. And I'm going to average across all the TRs within each scenes. And I'm going to get one representation that represents the activity patterns of voxels in this area for scene 1 and scene 2 and scene 3. Okay? Now, reactivation means that there should be correlations between event boundary representations and scene representations. So we're going to use representational similarity analysis, which basically means that we're going to correlate all our event boundary representations here with our scene representations here, okay? So this will give us a matrix of correlations. We also remove the main diagonal from this matrix, and the reason is that we don't want to measure the reactivation of the just ended scene. This diagonal represents the correlation between event boundary 1 and scene 1, event boundary 2 and scene 2. This is what has been found before, that we would find the reactivation of the just ended scene. We don't want to look at that. We want to look at information that is further away. So we're going to remove this diagonal. And now let's assume this matrix represents my talk again. So on one slide, you have an introduction to event boundaries, and then I'm telling you that there is replay at event boundaries and that there is replay of remote information. What we're going to predict is that at the relevant event boundaries, for example, when I'm talking about replay at event boundaries, at this event boundary, your brain should reactivate the relevant events, past events that should help you understand what I'm saying. And the relevant event here would be what event boundaries are, right? So at event boundary 5, we should find a correlation to event number 2 that holds relevant information. And when I'm talking about replay of remote information, we should see reactivation or correlation to the event where I told you there is replay at event boundaries, or what event boundaries are. Okay. Now there is a problem here, because these correlations might simply reflect scene similarities. The reason is that, as I said before, the fMRI signal is sluggish. Each event boundary is highly correlated with the scene coming before it or after it. So it could be that event boundary 3 is correlated with scene 3, and scene 3 is actually quite similar to scene 1 because it holds information about the same characters and events. So it could have a similar representation to scene 1. So this correlation between event boundaries and previous scene could just reflect correlations between the scenes themselves. And in this case, I wouldn't be telling you anything about reactivations. So we needed to control for this possible artifact, sort of a possible confound. So let's assume that this matrix indeed only represents scenes. There are no event boundaries. Everything we're measuring is the similarity between scenes. If this is a scene-by-scene similarity, it should be symmetrical. So this lower triangular part represents the past, and this upper triangular part represents the future. But obviously, if there is a correlation between scenes 5 and scene 2, we should also find a correlation between scene 2 and scene 5, right? Because everything here is symmetrical. So everything that happens, all the correlations that I can measure between event boundaries and past scenes, if this is all scene similarity, should be identical to all the correlations that I can measure between each event boundary and the following scenes. So what we're going to do, we're going to calculate this matrix, and we're going to deduce the future correlations from the past correlations. Everything that is symmetrical will be canceled out. There will be no scene similarity that will influence our analysis. And all we're going to be left with is the asymmetrical component that represents the reactivation of past events in event boundaries. Okay? So for each voxel in the brain, for each sphere that we create around it, we're going to calculate this matrix. We're going to calculate this reactivation index and assign it to the same voxel we started with. And we're going to do that across the entire brain to create a map. Okay? And this is what we find. So we're contrasting the past correlations with the future correlations. And this is what we see. This is the Sherlock dataset. On the left, you see an inflated brain. You can see a network of areas that show correlations between event boundaries and past scenes more than future scenes. You can see angular gyrus. You can see precuneus, which are areas of the default mode network. And if you look at this brain slice, you can also see bilateral hippocampus. Now, let's look at the 21st year data set. Very similar results. So we can see angular gyrus, we can see precuneus and retrospinal cortex, and again, we can see hippocampus. Now, I should also say that this data set has a lot of artifacts that we had to deal with. Basically, every result that I'm going to show you is going to be less strong in this data set compared to this data set. And although we have a lot of areas that we could interpret based on the Sherlock dataset, I'm only going to interpret whatever is being replicated between the two datasets, okay? So for example, you can see some sensory areas lighting up in the Sherlock dataset. I'm not going to talk about them. So I'm going to talk about precuneus cortex, angular cortex, and hippocampus. Finally, we can collapse our data together. we can pull all our data sets together and just to get an idea of the strength of this effect. And again, we see a very strong precuneus, angular gyrus, and hippocampus. And this basically means that default mode regions show reactivation of temporally remote past scenes at event boundaries. And I'm reminding you that these two data sets are so different than each other, that by the fact that we see this replication, it actually shows us something general about how we integrate information in our real life, because this is a very naturalistic experience. Listening to this audiobook is not very different than me telling you a story, and watching this movie is not very different than you watching this video of me telling you about my work. Okay, now let's talk about the anatomical scale of reinstatement. So far, so in rodent work, we can look at single cells. That's our resolution. It's very fine resolution. But in fMRI, we're looking at voxels, and voxels have many, many neurons in them. And so far, all the results I showed you were based on the voxel level. I was looking at one voxel and asking whether I can see reactivation in that voxel of previous information. But we know from other fMRI work of naturalistic experience that there is also correlation in larger parts of the brain. So there is this technique that I told you called intersubject correlations. And what they found is that if you look at scenes, movie scenes or story scenes, you can see correlations between the brain representations of different participants. So if this is a sphere in the brain and you can see the representation in different colors, then different brain would represent the same scene in a very similar way. Now, we can't really measure the exact same voxel across two brains. This is because our brains anatomically are very, very different. This is just an example. This is one brain and this is another, and they're quite different. Some brains have extra gyri, for example, or extra sulci. They have different shapes. And although we try to shape them into the same coordinate space or to the same MNI template, they are still very different even after this transformation. So when people take one voxel from one brain and another voxel from one brain and show a correlation, they probably aren't measuring the same voxel. They're measuring voxels that are in the surrounding of the real voxel. And this means that whatever effect they are finding, it is just shared across a functional region that is the vicinity of the voxel is a functional region that is shared across participants. And we wanted to know whether this reactivation that we found at the voxel level also has a structure at this mesoscale, at the higher spatial level. So to do that, we wanted to correlate between the brains of participants. So far, I've taken one participant, the brain of one participant. I've taken the representation and event boundary and at a scene, and I correlated the two, right? This is within a brain. What I'm going to do now, I'm going to do this correlation between participants. So let's say that this participant is going to be the one that gives me the scene representations. I'm going to take all scene representations from this participant. And I'm going to take the event boundary representation from the remaining participants in my group. There are only two here, but there are many more. So what I'm going to do, I'm going to take all these event boundary representations and I'm going to average them to get one event boundary representation for each event boundary. And this is actually very important because if I find reactivation in this method, it means that by averaging these different brains, I found a canonical or a shared event boundary representation, which would suggest shared binding. Because if we believe that event boundaries really do just summarize across different events, right? They just add up information at the event boundary and bind them together. This would mean that different brains actually bind the same information. They choose the same event to reactivate at the event boundaries. So this would tell us something about behavior too, right? Because the choosing of event is also consistent across brains. Also, unlike this previous method of correlating event boundaries and seeing within a brain, when we do that within a brain, we have massive problems of autocorrelations. Because this is fMRI signals, each time point is highly correlated with its surrounding. And by correlating event boundaries with scene, we are facing a lot of spurious correlations that are not related to reactivation. They're related just to these autocorrelations in time. And we did a lot of work and a lot of simulations and controls to make sure that the findings we have within a brain actually are not contaminated by this. But when we're using between participant correlations, we don't have this problem, right? Because we're taking signals from one brain and correlating them with signals from independent brains. So this is just to confirm that even if we didn't do 100% accurate job in controlling for these artifacts within brain, we have a replication that would show you that these artifacts can't bias this analysis in this way. So this is the same result that I showed you before. This is the within participant maps showing reactivation of past events at event boundaries. And now I want to show you the between participant events. And they are very, very similar. So again, in the Sherlock and 21st year dataset, you see angular gyrus and precuneus and hippocampus in the 21st year dataset as well, and in the meta-analysis. The signals are much stronger, obviously, and that is because we are benefiting from the averaging across participants. So we have a reduced signal to noise. But nevertheless, these two methods show us the same regions over and over again. And now, so I've shown you results. I've showed you that at event boundaries, we see these reactivations of past events. But is this effect actually specific to event boundaries, right? Maybe other points in time would show the same reactivations. What's special about event boundaries? So to answer this question, we are going to look at different points in time, both before and after the event boundary. How will we do this? So we're going to choose a point in time, say it's preceding the event boundary. And just like we did for the event boundary, we're going to create a correlation matrix. Here, instead of event boundaries, we're going to take one point in time before the event boundaries. And we're going to correlate these points with our scene representations. And we're going to do that for another point in time and another point in time and another point in time. We're going to have all these matrices for different time points that come before the event boundary. And we're going to do that for time points that follow the event boundary as well. And for all these time points, we're going to do the same thing we did before. We're going to take each matrix, we're going to subtract the future from the past into a number, and then we have all these numbers. Each time point now will have a number, and we can plot this number to show what happens to this reactivation as we move in time before the event boundary and after it. And I'm going to remind you how the bold signal looks. So when you are exposed to a stimulus, then you would see this initial dip, debatable, and then a rising bold signal with a peak at around 4 to 6 seconds, and then this drop. And now let's see what we see, not in the bold signal itself, but in the reactivation signal built on these matrices. So another important thing to say is that this method allows us to do a cross-validation analysis of our results, right? We have two data sets, they're independent. We can choose our areas independently from one data set and then test them on the data of the other data set and vice versa. And we can do that within brains and we can do that across brains. So the first result I'm going to show you is from ROIs, regions of interest, taken from the 21st year data set. And you can see these ROIs as black contours here, overlaid on the data, the group map of the Sherlock data set, right? So ROIs, as you can see, are quite different than the activations we see in the brain. This is the meaning of independence. And now let's look at the reactivation signals in these ROIs. Let's look at precuneus. And here the data is not shifted, right? I told you before that I'm shifting events by 40 yards. This is not the case here. So this is the event boundary. This is event boundary. This is these signals are averaged across event boundaries. So the red vertical line is an event boundary. And you can see the signal in precuneus rising until around 4 to 6 TRs, which represent around 4 to 6 seconds, as expected from the bold signal, and then a drop in signal. And you can see this for hippocampus, not significant in this comparison, and angular gyrus. And now we can take the ROIs from the Sherlock dataset and test them on the 21st year dataset. You can see the ROIs and maps here, but again, very significant results, rising and dropping for the precuneus, hippocampus, and angular gyrus. And this is within brain. We can do the exact same thing between brains. And here too, you can see very significant results mimicking the dynamic of the bold signal, but this is not a bold signal. This tells us that the point of time that shows us the most reactivation of past event is actually the event boundary. Because according to what we know about the bold signal, it should take around 4 to 6 seconds for us to see this peak. So there is something special about event boundaries. They do show the highest reactivation of past events. Okay, so reactivation, as I showed you, is specific to event boundaries, but is it specific to context? In other words, not in other words, let me demonstrate. So let's go back to my sound wave. Remember this when I showed you that on slide 4, your brain should reactivate at the event boundary of slide 4, your brain should reactivate slide 3 and slide 2 because these slides are relevant to what I'm talking about in slide 4. But your brain may not need to reactivate other bits of information. For example, the questions that someone may have asked and may not be relevant. So your brain has this task. There is a lot of information out there. It needs to choose the right information to bind together in each point of time for you to understand what is going on. So is this reactivation really selective for events that are relevant to our understanding? So the way we approach this is using a bag of word analysis. So what we're going to do, we're going to take the Sherlock movie and it has been transcribed. So not only the dialogues are transcribed, but everything that happens on the screen is just written down. For example, Sherlock is looking at Watson and says whatever. So this is going to be our basis for understanding which scenes are relevant to each other. And we also have the story itself from the 21st year, right? We have the text from the story. And we're going to take all these words from each experiment separately, and we're going to put all these words into a bag. Now, all these words, let's say this is the Sherlock dataset, are in this bag. All the words that have been described, has been used to describe the movie. And now we're going to look at each scene independently and ask which of all these words has appeared in this scene and how many times. We're going to do this for each scene separately. And under the assumption that scenes that are similar to each other, that share the same context, would also have similar words describing them, we could activate a bag of word model, just a correlation between these scene context representations. And this is how it looks like for the Sherlock dataset. You can see scenes that are highly relevant, highly contextually relevant to each other, and scenes that are less relevant to each other. You can actually see this more impressively when you look at the 21st year dataset. Remember that I told you you have narrative A and B and A and B and they're intertwined? So look at this checkerboard. This is what it means. One scene is related to the previous one, is not related to the previous one, but is related to the one before. It means there is this congruency and incongruency structure in the contexts of the stories. Okay, so we have these context similarity matrices representing the similarities between words. And we have our brain matrices, right? They are asymmetrical and they represent the reactivation that we see in the brain. And what we're going to do, we're going to correlate the past parts of these matrices and the future parts of these matrices. We're asking, if we see reactivations in the brain, would these reactivations more likely be of scenes that are related to each other based on their context compared to scenes that are unrelated to each other? This is what we're trying to measure. And we're going to contrast this with the future, again, to control for scene correlations. So this is going to be our index, this correlation between context similarity and reactivation in the brain. This is what we do for the Sherlock dataset. For the 21st year dataset, we have this problem of this checkerboard. If you think about correlating the brain reactivation with this checkerboard pattern, you can imagine that the checkerboard pattern will give you this jigsaw regressor. Congruencing, incongruencing, congruencing, incongruencing. And if we look at this regressor, it wouldn't tell us anything about whether there is reactivation within scenes that are related to each other. We're going to get a congruency effect. So to overcome this, we're going to use a time, past versus future, over congruency interaction. And let's see what we find. We're going to do this within participants and between participants. So for the Sherlock dataset, we find areas that reactivate past events that are relevant for each event boundary, contextually relevant, and they are the precuneus and angular gyrus and hippocampus that you can see here and other areas. For the 21st year dataset, because of a lot of technical difficulties that we discussed in the paper and happy to discuss if you want later on, we can see the precuneus, but it doesn't survive correction across the entire brain. And we can see this in our MEGA analysis as well. For the between participant analysis, we hope you have all significant results, again, for Sherlock in a network of brain areas for the 21st year in the precuneus, and again, in the MEGA analysis. And although it's encouraging to see that these effects survive a whole brain correction, if you think about it, the really critical question is, do the areas that we found to reactivate information in the previous analysis, are these the same areas that also reactivate information selectively? Do they choose which events to reactivate? So we have taken the precuneus, again, only because it's the only area that is consistent across analyses. And we independently defined it, and we find that across all data sets, we find that in precuneus cortex, those scenes that are relevant for your understanding are those that are reactivated more than those that are irrelevant. So again, this gives us the context modulation of this reactivation effect. So final event boundary. Let's sum up everything that I told you so far. I've presented very robust findings. They are replicated across two very different data sets. I've shown you that there is reactivation of remote past scenes at event boundaries, both within and between brains. Reactivation is specific to event boundaries. There is something very unique going on there, where at event boundaries, at these transitions between contexts, we bind information. This analysis of looking at the specificity in time to event boundaries also allowed us to replicate our findings, to do a cross-validation analysis, showing how robust they are. By looking at the correlation between brains, we find that there is a structure to this reactivation at the mesoscale. And by showing these effects, we can also interpret these results as showing a shared binding of past with present information shared across participants. Participants infer very similar scenes to bind together. Reactivation in the precuneus is specific for relevant semantic context, as I showed you in the bag of word analysis. So scenes that are more relevant would be reactivated. And just to sum everything up, this is an online mechanism, right? Because it happens while you're experiencing things, but it's executed by offline regions. The default mind network is mostly hypothesized to be an offline network. And it's also executed by offline mechanism, because replay has so far been involved in, has been proposed to be involved in coding events into memory, nothing that is currently happening online. So we proposed a revised functional role for replay and for the default mode network. Yes, it consolidates past memories. Yes, it can also use these past memories to inform future behaviors, but it can also interpret the present experience. And there are other papers that are now making this claim. So I want to thank my collaborators and for you for listening, for attending, and for inviting me to speak, and I'm happy to take questions.